Good day to you all. My name is Frederick Waas. I am from the Amsterdam UMC, and I will talk to you today on Metabolomics and Lipidomics, and really happy to be speaking to you at this DareFest 2022. So we will talk about metabolomics. So metabolomics is a name for measuring all metabolites in a given biological sample. For example, plasma or tissue or food. And all the metabolites together you call the metabolome. And by metabolites or molecules, we mean molecules that are quite small, so not as large as proteins or DNA, which are a lot bigger. So for instance, glucose, which you see here, is one of these metabolites that we can measure. But the metabolome is actually quite complex. It contains many different metabolites. So for instance, sugars like the glucose, nucleotides from which you build DNA, amino acids from which you build proteins, but then there's also lipids. So this part of the metabolome is also called the lipidome. So it's a subset of the metabolome. And we measure the metabolome using metabolomics to better understand how complex system works, to find biomarkers, and to understand disease mechanism. Metabolomics and lipidomics. So the fatty part of the metabolome is called lipidomics. So you measure all the greasy fatty molecules, which are really important molecules for your building your cells, but also you can focus on small molecule metabolomics, which is the molecules that like to be dissolved in water. So how do we do metabolomics? We use mass spectrometry, very powerful technique. But first, we generally separate the mixture of molecules from this complex mixture, which contains thousands of metabolites, by chromatography. So you probably have done this, where you dot a couple of pen dots on a filter paper, and then you put it in a suitable solvent, and then its solvents start to migrate upwards and separates out the different components that are found in this dot of the pen, and you see that it contains three compounds. So this is actually what this unit does. And then the mass spectrometer measures the weight of the molecule, or the molecular mass of the molecule, and by using different databases and different subsequent techniques, we are able to identify which molecule is what. So this mass spectrometer is used to characterize the molecules. And then if you look at what types of metabolomics we have, you have a metabolomics called targeted metabolomics and untargeted metabolomics. I want to go into that a little further. Targeted metabolomics, you measure what you can and already know. So for instance, you say, I'm going to measure acyl carnitines, or I'm going to measure fatty acids or amino acids, and look at those and see if I can find differences between, as you can see here, between young and old. So this is quite good established method. So it's easy. You make use of assays you already have and you already know. But obviously it's biased, because you won't find something new. Untargeted metabolomics, you measure everything that you can. You open up the detector and you measure everything that's gone by, and a molecule that you don't know you call a feature. So you actually, it's very unbiased. You might just find something new, which is obviously what we are into when it comes to research and development. But the downside is that it's actually quite difficult, both on the analytical part, but also on the data processing part. You literally risk drowning in your own data. So let's again think about why we want to measure all these metabolites and do this complex metabolomics. Well, one of the reasons is that the genome is very static. Once you have sequenced your genes, you know what your genetic makeup is, and this doesn't change. Of course, your genes code for proteins via the RNAs. You make proteins, and these work on molecules to build your cells. And if something is wrong in your DNA, this is reflected by what you do, your metabolite profile. And your metabolite profile obviously changes from young to old or from day to night. So metabolites actually represent of what's happening now. And as they can be easily measured in plasma and urine, they are the most proximal reporters of a phenotype of your genetic makeup or your current status or disease. And so they can be used as biomarkers for diagnostics, prognostics, but also to monitor treatment. As said, the metabolomics workflow is quite complex. Of course, you first have a biological question. Okay, for instance, if I have a Refsum mouse, what's the difference between a control wild-type mouse and a knockout, a Refsum mouse? So you set up an experimental design, you have a protocol, then you do all your sample collection. For instance, I say, I will collect the liver, you collect these samples, and then you do the measurements of the data acquisition. This gives you this raw data. And then you have to do this data processing, which really is quite difficult. You have to pick out all the peaks, then put a name to the peak so that you end up with a list of metabolites. And then obviously this data analysis part is not complete until you put it into context of metabolism, so in pathways or in ways that say something about the context and answer your biological question. So let's look at our metabolomics platforms as an example. We use for our lipidomics platform on one sample in our analyze, we separate it on two different columns with two different properties to get a better picture, and we measure both in the negative and the positive ion mode because molecules can be ionized either positively or negatively, and some are not ionized in the positive mode and others are not ionized in the negative mode, so you get a more complete picture if you do both. And we do this on a high resolution mass spectrometer, so very good one. And for metabolomics we use for one sample, one column, and measure again in the positive and the negative ion mode, and we use different machines because of analytical reasons. And it's good to realize that in one of these files you can get up to 30,000 features, so up to 200,000 features you have generated on one sample, and you have to process all this data. And for this we have a very efficient and reproducible bioinformatic pipeline, which eventually generates all these Excel documents and graphs and does statistical analyses for you. To give you an idea about this bioinformatics pipeline which we have built, you can see here that we built a special program to look at the different lipids. So if each dot is a lipid, so here you're looking at two columns, so the negative and the positive ion mode, and each dot is a feature. And by using our analytical knowledge and internal standards, we, for instance, are able to say, okay, these are all triglycerides, and this is the triglyceride cluster. And this program, which we made ourselves, helps us to annotate the lipids, so to put a name to a feature. And then we have a statistical analysis pipeline, or that takes all the list of metabolites that we have created and generates visual or graphs or heat maps or all kinds of graphs that help the researcher to interpret this huge amount of data, which is obviously something that is nice to have in an automated way and really helps you to understand your data. So just to give you an indication, because you might wonder, okay, how many lipids are there actually? Well, in the current lipid maps database, which is available online, they say that there are about 50,000 unique lipid structures. So this is one of the biggest parts of the metabolome actually is the lipidome, because there are so many different lipids. This makes it quite complex. So I hope with this brief overview of metabolomics and lipidomics I have given you an idea of why we do it and how we do it. And I want to end with just 2 slides on preliminary results which we have generated on the Refsum mouse model. Using the Refsum mouse, we made 4 groups of each 6 animals. So a wild type mouse with a normal diet or a phytal enriched diet, and the same for the Refsum mouse. So here you see the color coding for this graph. And we did liver lipidomics, and yeah, that yielded about 2,400 lipids that we could annotate, that we could identify. And just to give you an idea on the results very briefly, and then in the next slide I will show you more specific results, but this is a general view of looking at such an experiment. You can do what you call a PLSDA analysis, where in this 2D plot, yeah, I could say that it's like it's a map, that if you have 2 groups that look like each other, that are similar to one another, they group together, and the further the difference between the groups, the more different they are. So what you see is that the wild type groups, so with the control diet in yellow and with the phytal diet in green, are quite close together. That the Refsum mouse without a normal diet is already quite well separated from the wild type groups, even on phytal. But if you give the Refsum mouse phytal, then the lipidome changes incredibly, and it is completely separated from the wild types. So this is one way of looking at the experiment. So this, you can conclude that indeed there are large differences occurring, and then we zoom into the specifics in the next slides. One thing that we could conclude from this experiment is that many lipids incorporate phytanic acids. So phytanic acid is made from phytal and then is incorporated into what we call complex lipids, so lipids that contain more than 3 building blocks. Fatty acids obviously normally are a building block in that respect, and instead of normal fatty acids, phytanic acid is incorporated into them. So we use this lysophosphatidylcholine, LPC, as an example, and this is LPC with a phytanic acid. So this is normally in the wild type situation very low, and even if you give a wild type mouse phytal, it will make phytanic acid, but it will not incorporate much more of phytanic acid into complex lipids because it's broken down. But in case of the Refsum mouse, you already see that the levels are much higher, and if you give phytal, then the levels of phytanic acid containing lysophosphatidylcholine are even much higher. And this is a general phenomenon that we saw for a lot of different lipid species. So this could be a new biomarker. This could be relevant for pathology of Refsum. I really think so, that it is. So this gives you new ideas where to look, but also hopefully new insights which will lead to new therapeutic options. At least that's the hope, obviously. So, and with this I would like to conclude my presentation on metabolomics, lipidomics, and a sneak preview on the Refsum mouse lipidomics. And thank you very much for your attention.